If you wanted to be like a super terrorist, what are the things that you would do?
Generally, like when we're talking about like bioweapons, the reputation is like a poor man's nuke. If you generally know your way around a lab, it's really not that tricky to encapsulate the purified virus, or just to spread the purified virus, right? You could have a terrorist walk into John F. Kennedy Airport carrying a little tube of Ebola, spread it across the handles in all the major bathrooms. You would never know that it was him. In 2001, after the 9/11 attacks, there was a couple of reporters, the journalists and politicians who received letters in the mail. In the letters, when they opened them, there was this dry powder. What they realized is that powder was actually anthrax. The federal government, specifically the Department of Homeland Security's policy, is that BioWatch does exactly what it was intended to do, which is detect the threats that we know how to counter.
It doesn't detect the things that we don't have counters for?
Exactly.
On purpose?
On purpose.
Today, I'm super excited to interview Jake Adler, the founder of Pilgrim. Jake, when are we going to get the first super soldier?
So it's one of those things where, I've really believed since, the creation of Pilgrim, but even before that, before we can go about like enhancing warfighter capabilities, we want to first opt to restore. So, as we look at the battlefield, we're kind of moving away from this world that we saw in the global war on terror. So in Afghanistan and Iraq, where we had counterinsurgency operations. So warfighters were deployed into these environments that were readily accessible. We could easily evacuate them and bring them to military treatment facilities or bring them to proper treatment or therapeutics, to a world where warfighters are going to be dispersed and distributed across this massive battle space with, frankly, very limited access to the individual warfighter, to the medics. We're expecting, highly contested airspaces. So it's one of those things where warfighters are going to be deployed into combat without the relevant medical tools or infrastructure in place needed for their survival. So it's, before we can kind of approach the question of enhancing the warfighter's ability, we need to ensure that they can survive and ideally thrive in conflict environments. And that's really been the underpinning component of what we're building at Pilgrim, specifically around like a next generation hemostatic. So, something I've tested on myself, but notably something that can rapidly stop the bleed and help regenerate tissue. And the relevance here is, we're moving away, like I said, from that period of time where we could bring a warfighter back and properly treat them, to a space now where you have situations in Israel or Ukraine where warfighters are being deployed and are actively injured, and tourniquets are being deployed and left on for far too long. So obviously, there's a really clear opportunity to build a solution that actually accommodates the movement towards the future conflict environment that we will be encountering. And that's really been the driving force behind a lot of the stuff we do at Pilgrim. So I think on the point of enhancement, while I'm a really big Captain America fan, I think, the immediate reality, at least, is there's a lot that has to be done to support the warfighter before we can move towards trying to augment or elevate their capabilities above our adversaries.
Yeah. And for the first product that you guys are creating, it's just literally like a patch that you put on you're bleeding, you put it on your skin, and it stops the bleeding, right?
So there's probably warrants a little bit of backstory, specifically around it, and a little bit of context around what kind of already exists. So if you've seen Black Hawk Down, or the events that happened in Somalia or Mogadishu, around that period of time, there was a surgeon named John Holcomb. And he was one of the army surgeons actually at Mogadishu, and one of the people who was treating the warfighters as they were getting injured. And his recognition was largely that these warfighters were just bleeding out. Like we didn't have a solution to stop their bleed, right? And
how many people bled out?
It was a very significant amount, but it's one of those things where it's been a prevailing issue where tourniquets can only be applied in very specific areas of the body. So generally, when we're talking about tourniquet placement, when we're talking about junctional regions of the body, so the groin or the armpits, you can't truly apply a tourniquet there. Now, we have been working towards building like junctional or truncal tourniquets for truncal hemorrhaging or truncal hemorrhages. But where we are today, generally, tourniquets are not sufficient solutions for that.
Is that because there's like right underneath?
Exactly, right? Well, not only that, but it's hard to physically create pressure there. Also, think about the abdomen. Trying to create pressure on the abdomen when the abdomen can easily compress is not very effective, right? So that was kind of the early inspiration for the creation of the early hemostatic agents. When it first came around, there's a guy named Frank Hersey, and he had a company called Z Medica. And Z Medica did I think they were doing medical he was doing like nitrogen generators, basically like air filtration units. They weren't doing anything adjacent to blood or hemostatics or trauma or military, right? And the original solution that he actually developed was something leveraging a mineral called zeolite. And he actually he discovered it just like really serendipitously because it's typically used in like these filtering units. And what he would do is he would cut himself shaving and he would pour zeolite on his wounds so it would like stop the bleed, right? And he's like, this seems like it's something really interesting. And this kind of this converged with the period of time where John Holcomb was really interested in this idea of having a gauze that could more meaningfully stop a bleed, right? The challenge was that generally as it comes to trauma care and it comes to working with medics, these field medics are extremely risk averse. So they understand gauze really well. They understand tourniquets. We've had tourniquets for almost close to 2,500 years now. They're first deployed by Alexander the Great, and they haven't really evolved much since. I can actually talk about that.
Let's go into that.
So I mean, it's one of those things that's really crazy. Where throughout history, you know, there's been instances. There's even this one incident where a warfighter just was like this group of warfighters were never trained on how to even use the tourniquet. So they found an individual, like a soldier, who actually died from a hemorrhage and had a tourniquet in his pocket. Like he just didn't even know how to apply it, right? Or we're like looking at the current conflict right now in Russia, Ukraine, where we're seeing people putting on like bilateral tourniquets. So basically putting them on both legs and leaving them on far too long. So if you leave a tourniquet on for too long, it can not only necessarily lead to amputation, but it can also readily lead to death, right? So it can cause an inflammatory response within the body that can trigger really the body to break down. And this actually recently happened, I think, to a 19-year-old, which is very tragic. But point is,
and how long do you have to leave it on for it to
so generally, like, you don't want to leave them on beyond 6 hours, or like maximum like 12 hours. Right now, you know, I was recently at a really large, you know, military medical conference, and at this event, we had people, like I said, coming in from the IDF, from the Ukrainian forces, and you know, they were describing people leaving on tourniquets for days at a time. So it's one of those things where,
is it just a lack of knowledge, or?
I think lack of knowledge is a big part of it. You also kind of have to recognize the solution that we're deploying with. So the leading tourniquet that we deploy warfighters with in individual first aid kits, or what we call IFAKs, is the CAT tourniquet, which is sort of the pride of North American Rescue, which is a really big integrator for first aid and trauma care components. And the CAT tourniquet, we actually have one over there. All right. So this is the Gen 7 of the CAT tourniquet. Now, this has been around, I think, for about a decade or about 2 decades now. And this solution is obviously fully manual. Now, for it to go from Gen 6 to Gen 7, they only made like 2 additional differences. So I think they added this little red tab here, and then they added like the manual spinner here for compression, whatever the term is, the proper term for this. We'll call it a spinner. But point is, for it to go, for them to actually integrate the red tab and like the manual spinner and the lock, I think took like 5 to 6 years. Because it has to go through this really intense process within the Defense Health Agency. The specific approach relies on involvement from the Joint Trauma System, specifically COTC, or the Committee on Tactical Combat Casualty Care. And you also have to work with the Institute for Surgical Research, which is a part of the Army, and also has some involvement from the Defense Health Agency. But it's one of those things where, like I kind of mentioned, these combat medics are incredibly risk averse. They want to field solutions that they recognize, that they're aware of. So like in the early days of the global war on terror, we weren't deploying with these. People were getting deployed with basically cloth and a stick. So they would be basically making, like, you know, homemade tourniquets
Incredible.
to accommodate, you know, severe hemorrhage. But what eventually happened is, you know, you had John Holcomb and you had a lot of people who start to recognize the potential of a gauze or some agent that could stop the bleed. And that kind of leads back into Z-Medica and zeolite. Now, zeolite was one of those sort of offhand ideas that kind of really initially worked, but got a lot of pushback, notably because zeolite is a concentrating agent. And what that means is when you applicate it to the blood, it basically concentrates the coagulation factors and sucks out the water. The problem is when it sucks out the water, it traps it in the crystal lattice, and that process is incredibly exothermic. So it releases heat. And what would happen is we would be deploying zeolite onto warfighters, and we'd cause 2nd or 3rd degree burns while we were stopping the bleed. And
How many people did this like happen to?
Oh, it must have happened to hundreds, or if not thousands. Like a lot of people were deploying with like.
Would you just do like permanent scar tissue and stuff?
Well, I want to be clear. Like, zeolite saved lives. Like, it definitively was a solution that was effective for stopping a bleed. And it came at a time where it was definitively needed, right? In those early days of the global war on terror, it was a new conflict. Tourniquets were kind of letting us down. We needed a solution that would actually accommodate or support the warfighter who was going into these environments, and ideally, stop the bleed without as much education on how to properly apply the tourniquets or having to be concerned about how long to leave the tourniquet on for. So initially, Z-Medica came up with a zeolite powder, and they labeled it Quikclot, which is sort of that early solution. And it was one of those things where it had to go, obviously, generally, because the DoD tries to be competitive, it went up against a few different solutions. One of those is from a company now called Tricol Biomedical, and they had a product called Hemcon. Hemcon actually looks something similar to this. I actually think Cellox is now owned by Tricol, but this is a chitosan-based hemostatic. So basically, they took shellfish and they crushed up a lot of shellfish, like the shell, and embedded it into the gauze. Now, this is not a true hemostatic because it simply helps with the thrombin formation, like the clot formation, but it supports the overall hemorrhage control. The problem is, if you are allergic to shellfish, you can't use this product. So
is that like how many people are allergic to shellfish?
I don't think it's like a severe issue, but it is like one of those underlying things. It also isn't as effective as Quikclot, but in the early days, these two were going up against each other, and Quikclot came through ONR, so the Office of Naval Research, and Hemcon was driven through mostly by the Army. And it was one of those things that you could pull it out of like Edison's biography, where you literally had the Army go up on stage and they would take a styrofoam cup and they would pour water in the styrofoam cup, and then they would pour in the zeolite powder and it would burn a hole through the styrofoam cup. And what they were trying to do is demonstrate like the overall thermal effect, using zeolite would cause, like the exothermic reaction. And it was one of those things where they're trying to undermine the credibility of the product, even though Hemcon had really poor clinical data supporting its hemostatic effects, right? So eventually, through a lot of like guerrilla style work, Z-Medica managed to kind of get Quikclot proliferated across the DoD and the Joint Forces, but it definitely was a really severe challenge for them. And really in that process, I think in 2020 they were acquired for about $500 million by Teleflex. Now, Teleflex is a really interesting organization. They do a lot of different medical device development and manufacturing. But what's really challenging about a company like Teleflex is they're one of your traditional like beltway contractors, where they are very well established. They're a big medical tech company, and they've hit a point now where they're so comfortable with Quikclot as a solution that they fired most of their business development team. So at the largest conference, like the one I just went to, where you had people from Tricol, you had people from Gauze, you had people from Blood Stop and Wound Stop, like these new next generation hemostatics, Teleflex didn't even show up. And yet they're the largest supplier and manufacturer of hemostatics today, right? So, between like 2017 and 2021, the DoD procured like 650 million IFACs, or 650,000 IFACs rather. And each of those contains at least one hemostatic agent, if not two, right? So, Teleflex is pulling in real revenue, but yet they haven't changed their product. They haven't evolved in really close to about a decade and a half now. And it's one of those things where they're almost in their lazy boy recliner, like
just sitting on the laurels.
Really just sitting on their laurels, right? So it's one of those very clear vacuums where I think there's a clear opportunity to establish a wedge in creating not just a solution that stops the bleed, but something that would actually accommodate tissue regeneration. And this is relevant because when we're talking about the movement towards these large-scale combat operations where medics might be expected to be in combat for weeks at a time, where the warfighter is going to be injured on the front lines, and if they're injured in a forward deployed setting, and they go down on the X, they have to effectively maintain, or that unit has to establish fire superiority before they can basically, remove that troop and bring them to proper treatment, even if they have access to treatment options, right? But if they don't, which is likely going to be the reality, we need to have something that would actually expedite the return to duty of these warfighters. The definition of a casualty is changing, not just from somebody who's been severely injured or unfortunately died, but somebody who can pick up their gun, right? So if you can't pick up your gun anymore, you're going to be classified as a casualty, right? And in the future conflict environments, when we're seeing the emergence of novel offensive systems that are like drones. And we can kind of get into that a little bit more too. But generally, when we're seeing the usage of these weapons that can effectively hit units in a much more of an intimate way, something we haven't really seen in past conflicts, we really need to ensure that we can maintain a effective warfighting force for a longer period of time, right? So when we're looking at like major conflicts and we're thinking about what actually wins wars, a big part of it has to do with how long we can endure, how much we can bring to the warfighter, how much from a logistic standpoint, how much from a medical standpoint. And I think, where I'll end it here is like, it's generally pretty unfortunate because really, like at its core, a warfighting force is only as effective as the medical support that they have, right? And you actually can even look to a psychological level, and warfighters who don't have medics and units are generally lower in morale because they're not going to take as much risk if they don't know if there's somebody nearby to treat them if they get injured, right? So it's one of those things that I think is highly relevant. I think Pilgrim is probably one of the only defense technology companies today betting on the human over the machine. And I think it's a bet that I think is going to be enduring and prevailing in many, many conflicts to come. One thing I actually forgot to mention is this, which is actually a solution coming out of Israel. This is actually an electronic tourniquet. So it's the first of its kind. It's one of those things that on face value is like really cool, but then you realize how stupid it is because it has no sensors. So you would ideally want an electronic tourniquet to be able to monitor the pressure and monitor like blood pressure, blood oxygenation, determine whether or not you're cutting off, critical blood supply that might lead to an amputation. And this does none of that. So initially when I got this, I didn't really know how powerful it was. So I just pulled off the red tag and I wrapped it around my leg. And I had to like almost sit there like sawing it off to like actually get the tourniquet off. But the one thing it does have, fortunately, is like a timer, which is really relevant for people to like come in and or for like medics.
So in case it like completely cuts off blood circulation, like maybe it'll come back on in 5 minutes.
No, it's more for like a field medic to be able to keep track of how long the tourniquet was on for. The problem though is imagine, we're looking at mass casualty events. Like mass casualty events are going to become an operational norm. So if you have, if we look at October 7th as an example, or you look at like what Magen David Adom was doing, which is sort of like the leading ambulance provider in Israel, and sort of what the support they had to provide to people, war fighters, but also civilians. To have this, solution deployed and having electronic tourniquets appears on face value to be superior. But unless you have a meaningful way to keep track of the time across thousands of tourniquets, maybe at one point, it isn't really relevant, right? So, this is a really compelling concept. I think there's a lot of room for improvement on what they're trying to build. And for a concept like this, they're also looking for procurement pathways. They're looking for ways they can get a hold of ISR and kind of establish a presence in the DoD, especially considering they're a foreign contractor. But I think it's a really good step in the right direction.
Yeah. So for this like first product, how are you thinking about kind of getting it, I guess like taking kind of the minimum number of days in between today and when a bunch of people are using them?
So generally, when we're talking about medical device development, and I'm going to start off in like a more of a civilian-oriented context. So, a traditional medical device startup might go out and raise, a certain amount of private capital, do the relevant prototyping. They'll have to likely do some variants of preclinical, so animal modeling or animal models, and clinical validation to determine the overall safety and efficacy of the product. And throughout that process, they're likely going to be doing a concurrent review or clearance or approval process with the FDA. Now, this really comes down to the risk profile of your technology. So if you're building something like Neuralink, right, then you're going to end up in the class 3 medical device category. So you're going to require something called pre-market approval or a PMA. And before you kind of go through that process, you also have to get something called an IDE or an investigational device exemption, which enables you to even put it on a human or to surgically implant it in a human in the first place. Just to kind of flag post it a little bit, when we're talking about medical devices, there's 3 distinct classes. The first might be, class 1 is going to be your bandages, your gauzes. Class 2 is going to be like condoms. Most people don't know that those are medical devices. I think contact lenses and CGMs also fall in that category.
So is there like certifications for like, condom testers, people going in factories and saying like, yep, this is a good condom?
So it's a good question whether or not it needs CGMP. I wouldn't, it's actually something would be maybe interesting to look into. CGMP stands for current good manufacturing practices. And that kind of complements good laboratory practices.
I'd imagine that for condoms that people would want that.
Yeah, you'd hope, right, for contraception. But who knows? But anyways, it's and then the third class is going to be your PMA, or rather your class 3, which requires PMA. So generally, when we think about new technologies at Pilgrim, we don't do drugs. So we don't touch therapeutics. We're not going through the investigational new drug route or new drug authorization, which on average an NDA can cost anywhere from like $900 million to $3 billion, right? So we focus much more on the medical device side of things. And in that medical device, realm, our sweet spot is like class 2 devices. So in the class 2, you can take two distinct approaches. The first is something called 510K, and the second is called de novo. 510K is basically a clearance process that's based on the assumption that a product has already gone through de novo. It might not necessarily be your product or be a product you've made previously, but let's say your competitor built this product, they put it through the relevant clinical testing, they brought it to the FDA through a de novo process, and then they established something called a predicate. So a product that the FDA recognizes, they understand the risk profile of it, and then you can come in as this competitor and say, well, you guys already looked at this other product, right? We built the exact same thing. You already know the risk profile. Let's just clear a product as well. And that can be very, very fast. So I think 510K, there's different prices depending if you're a small business or if you're a larger business. And I think that comes down to employee count and revenue. But generally, I think it ranges from like, I want to say $5,000 to like $30,000 to go through that process. And I think a de novo is like about 50 or 60. But don't quote me on those numbers. They might have changed. They change every single year. But point is, there's these two really distinct pathways. So in terms of how we approach device development, it's a little bit different. And this is sort of I think really the key underpinning of like what we're trying to build at Pilgrim, which is on face value, trying to create this new generation of military medicine. But really the subliminal point here is how can we more effectively translate technologies, right? So, my core thesis going into building Pilgrim was that, we have this abundance of really compelling concepts that are sort of languishing in academic literature, like orphaned, don't see really the light of day, right? And, researchers will continue to pursue, this wide spectrum of research out of their own vain curiosity. But when it comes time to actually take that research and turn it into a meaningful product, they sort of toss their hands up in the air or poo-poo the idea overall, right? So it's one of those things where, there's probably like 30 to 40 years of really incredible technology that's sort of just sitting there. And what we do at Pilgrim and sort of what I've been really good at for most of my life is piecing little things together. So even this thing you see behind us is our Argus platform. So this is a near real-time, fully autonomous biosurveillance platform that's intended to detect the presence of pathogens in the air and effectively do collection, detection, and characterization. So we can kind of a little bit later on go into like the current paradigm of solutions, but for something like this, the impeller unit's actually using a Dyson, like a Dyson fan unit. So I broke down one of the Dyson fans that you can get at Target, and I stripped out the impeller, and I integrated that impeller to actually serve as the intake mechanism for basically pulling in the virions or the virus, like the viral particulates in the air, right? And then additionally, all the other hardware is right now this is mostly just makeshift stuff. But my point is like it's kind of taking and bridging these different concepts together, bridging different technologies.
And what is that actually going to test for? So what different viruses?
So the really critical piece here, and I mean, we can kind of jump into this. I just don't want to get off track, but the current solutions in the market or kind of what we have deployed at a national level today is still heavily reliant on like PCR technology or polymerase chain reaction technology. So if you ever, I'm sure you did, but if you guys recall during COVID, or if you recall during COVID, like the nasal swabs you would have had to do, those were a PCR test, which you're probably familiar with that term. My point is that a PCR test requires a priori knowledge or priori knowledge, which means that we need to first have understood the threat or first sequence the threat to then establish the relevant primers to then actually conduct PCR. And this is kind of where the challenge comes into place, is that PCR can only detect the known threats. It can't detect anything emerging, right? So what we're building here is something completely different. So most of what you're seeing is the intake mechanism. So basically we're pulling in these viruses. We're doing like a special process to basically collect them to like a very specific plate that basically breaks down the outer layers of the virus. So you have the capsid and the viral envelope. Basically the capsid is generally a protein, or rather the capsid is protein. I think the viral envelope is the lipids. And effectively what you're doing is that's correct. And what you're doing is or what this device is for, is to break down both those outer layers, basically suck out the nucleic acids or the genetic material. And instead of doing PCR, which is basically the test I mentioned, where you need to have that prior knowledge, what we're doing here is sequencing. So we can basically, it's really interesting. But if you remember that game you played when you were a little kid with that big wood crate, and you would have like this very specific shapes, and you can put blocks with specific shapes. We do something very similar, but at the near atomic level, and this is called nanopore sequencing. So basically, what you can do is you can have a protein that will basically grapple onto the strand of DNA or RNA, depending on the virus. So you might have double-stranded or single-stranded DNA, or likewise for RNA as well. And basically, you can separate it. You take the nucleic acid, and you have this little pore, and you can feed the nucleic acid through the pore. And it sounds like it's literally like the game where you're putting the block in, and based off of the base that's going through the pore at one time, there's a change, and I don't want to get overly in the weeds here, but there's a change in the ionic resistance. So there's a general disruption of ionic current that indicates which base is actually in the pore. So you can basically have a mechanism that can do near real-time, extremely rapid sequencing without requiring something that is as big as this container over here, and would enable us to effectively be able to understand the threat that we're collecting in real time. So the reason this is relevant is if we're talking about something like H5N1, right, where like the avian flu that we're seeing right now, where we have like, we've had to kill like roughly 175 million chickens. I think 170 million chickens at this point. The cost of eggs have increased well above like 300% to like, I think $6 a carton at this point. But H5N1 in humans has a mortality rate of close to 50%. So it's a bit of a coin flip whether you're going to live or die. And right now, the H5N1 strain that is predominant across the country is largely enzootic, which means that it's mostly within the animal population. So it's not transmissible in humans. Researchers, like I think 5 years ago, determined that it only requires 5 point mutations, so 5 mutations in bases, for it to spill over and become zoonotic, or to become transmissible in humans. Now, if you're relying on PCR technology, you wouldn't be able to necessarily determine or see that distinction. And that's incredibly relevant when we're talking about trying to do some variant of real-time detection or improved detection, where we want to be able to tell whether or not you're looking at a strain or mutation of H5N1 that is transmissible in only chickens, or has actually now spilled over into the human population as well, right? And the current solutions don't enable that capability. There's also a wild set of other challenges involved in the current solutions we have deployed. Even
Do you want to talk about those?
Yeah. So there's sort of two distinct angles you can think of. Notably, you can start off kind of from the national security angle. So in 2001, after the 9/11 attacks, there was a couple of reporters, so journalists and politicians, who received letters in the mail. And in the letters, when they opened them, there was this dry powder. So initially they probably thought like, whatever, right? Like it's a prank, right? And roughly 5 days later, they started to develop symptoms. So they started to develop fever, they started to develop diarrhea, they started to have irritation in the lungs. And what they realized is that powder was actually anthrax. So it was basically the anthrax spores. And basically in the letter that they would open, it said like "Death to America," like in like the most like terrorist looking writing you could imagine. There was a long string. And point is, initially, obviously we assumed that it was like related to Al Qaeda or related to some adversary. And as we kind of continued the investigation, we had to actually close down a couple post offices because the letters were obviously used or deployed through the Postal Service. And I think roughly 5 people died, 21 people were injured. But
like just at the post office?
No, generally from the anthrax attacks. It's called Amerithrax. Most people aren't aware that this occurred because it kind of is overshadowed rather by 9/11. But point is, obviously it was a really severe attack, and it was one that we just didn't really have any mechanism to prevent. So as we can kind of continue the investigation and we actually looked at the spores, we recognized that they had some level of familiarity. So they weren't actually novel. They didn't look engineered. And when we actually put a little more time into it, we realized that the spores actually originated from a sample at USAMRIID. USAMRIID is the Army Institute for Infectious Diseases. So USAMRIID's sort of been the key player involved in the U.S.'s role in biological weapons and chemical weapons through the Edgewood Arsenal, through Fort Detrick, and through generally a few of the different installations they have. But point is, we recognize that the origin of these spores came from what we call RID, or the Institute, which is USAMRIID. And as we kind of did more research, we thought like that's pretty odd, right? And we recognized that it was actually an Army officer. And interestingly enough, it was an Army officer who I believe was leading the development of the anthrax vaccine. So to gather or garner demand for his anthrax vaccine, he proliferated an anthrax attack,
wow,
killing 5 people and injuring 21, just to create that relevant demand to drive appropriations towards what he was developing.
Has there been any other cases like throughout history or even in the US of people doing something similar to that? Like,
so there's another theory. Again, I'm going to say theory, but there's another theory around USAMRIID's involvement in tick-borne Lyme disease. So there was a researcher who was actually working on trying to determine what viruses or pathogens they could embed with ticks as a means of using them for as a biological weapon. So this actually relates to something Japan did in their Unit 731, and this would have been during World War II. They had something called the Oogie bomb. And it was basically the ceramic bomb, and it would have flies or fruit flies that they would coat in viruses, or they would just be carrying the virus.
They just drop that?
And it also food. And basically they would drop that bomb onto a city or onto a region, and what would happen is the fruit flies would rapidly proliferate, and what would happen is the food would attract rats and would attract other small creatures, and the fruit flies would kind of gather around those rats and infect a bunch of like the smaller animals in the city, and those rats, as you know through like the Black Plague, were one of the leading causes of transmission, right? So it's one of those things where
how many times did this happen? Like how many cities were this dropped on?
So I'm not sure of certain occurrences of it. They were really nasty. Like units. So the people leading up to 731 actually got, at least some of them got immunity after World War II for sharing like a lot of information about what they were building. If we kind of look, like there's somewhat of a modern parallel to what the CCP is doing right now, and along with the Russians. So like even recently, there was this instance of a reporter who used Planet Labs, which you're probably familiar with, and they actually surveyed they kept track on a couple like the major Russian bioweapon facilities, and they saw a movement, and actually we're seeing now that Russia is actually modernizing one of their high containment BSL-4, so the most severe pathogens you can imagine. So right now, and I don't want to get sidetracked, but smallpox, which is probably one of the worst viruses in history, one of the worst pathogens we've encountered, basically causes these big boils to form on the body. The reason it's so bad is smallpox, the actual particles travel like smoke. So for example, there's an instance of, I forget where it was specifically, but there's an instance of a hospital where a patient had smallpox in one room, and basically, I think on the opposite side of the hospital, somebody also contracted smallpox.
Just through the like AC vents or
through
something?
Through the AC vents, and generally through the windows, like leaving windows open. But my point is that generally, when we're talking about these threats, BSL-4 facilities would be containing variola, which is smallpox. After smallpox was eradicated, we basically kept 2 samples. So it was sort of like a Cold War era kind of thing. But one of the samples went, or it was, I think maybe before, but one of the samples went to the Vector Institute in Russia. And then the other sample went to the CDC in the US. Now, we're not confident generally from an intelligence standpoint, from what I understand, that Russia has not removed that smallpox.
And why did we give it to them?
That was sort of part of the deal. It was like sort of like the final, like it's like like the seed vault. Are you familiar?
Yeah.
It's like sort of like the seed vault,
except for like an extremely deadly virus.
Well, I think that the premise is that if we started to see the proliferation of it again, then both of the major superpowers had the relevant virus to then begin to work towards maybe developing a new vaccine. Now, there's also a report that I think like the World Health Organization, I think it was them or the UN, they had this big trove of variola vaccines, and we don't get vaccinated for variola anymore because it's been fully eradicated, which is also an interesting story how they did it. But it's very similar to sort of the strategy with Ebola as well. But if you look at your parents' arm, if they're assuming they're Canadian or American, you might see a scar on their left arm. It's like this weird little bruise. That's actually from the variola vaccine. So they basically stab your arm a few times. Like they had a needle that would stab your arm, and then they would inject you with the vaccine just to get to the relevant immunity. But we don't do it anymore. But point is,
stop?
Why did we stop? Mm-hmm. Oh, because it's been eradicated. So it's one of those things where having to do that obviously causes like mental scars on people to stab them, but also producing the vaccine for a threat that isn't relevant is irrelevant. My point is that we actually had the World Health Organization, they have this massive trove of variola vaccines, and I'm pretty sure that they didn't want to pay for the refrigeration costs, and they just dumped all of them. So now the U.S. has a policy that per every one citizen, we have a variola vaccine in storage somewhere, which is obviously very great. But you got to have come to this question of like, and look, I got 5 COVID vaccines, and I can walk you through them. I even got the Chinese ones too.
Wait, why? Why'd you get all of them?
I was international at the time, so
Interesting.
I got Sinopharm, and I got Pfizer, and I got Moderna.
Did you have any side effects?
The worst was from Sinopharm, so the Chinese
what was that
one.
like?
Just incredibly lethargic, sick. And actually, there's a reason why that happens is because they're giving you an extremely large dose of mRNA, largely because the delivery vehicle they're using. So this wasn't the Sinopharm one, but Pfizer, Moderna, both were mRNA vaccines. The mRNA vaccines need a vector, or they need a system to actually deliver it to your cells. And what they use is something called a lipid nanoparticle, which is this little fatty particle that you can embed based off charges, basically mRNA, or generally you can embed nucleic acids, but it works best with mRNA. And the problem is that lipid nanoparticles, they reach the cell membrane or the cell wall, and they basically cause the cell to form what we call an endosome, or basically a little vehicle that can bring in this little particle carrying the mRNA into the cell. The problem is that that vehicle, the endosome, is incredibly acidic. So it's very low on the pH scale. I think it ranges like a late stage endosome is like 4.3 in pH. And what happens is it disrupts the lipid nanoparticle. And if that lipid nanoparticle can't break out of the endosome, then the mRNA is broken down by enzymes. So I think roughly, basically, if you got it, did you get a Pfizer or Moderna vaccine?
I got, I think, Johnson & Johnson and Pfizer.
Okay, so the Pfizer BioNTech vaccine used this solution, and basically, what we call so that first process of it getting into that vehicle is like 80 to 90% efficient. The process of it escaping that vehicle is like 5% efficient. So they had to give you like 50 micrograms of mRNA. I think it was
like 20 times as much as you actually like need, or
it was a significant amount more than you would need because of the limited efficiency in actually getting it into the cytosol or the cytoplasm of your cells, which then enables the actual spike protein to form, and that creates the general immunity, right? So back to my point on smallpox, though, those are contained, or that would largely be used in a high containment facility or a BSL-4 lab. We're currently in a BSL-2 lab. So like today, I just received like rhinovirus, which is the common cold, and an H1N1, which I can bring if you want to see it. But it's one of those things where also we can handle recombinant proteins. So like in-house here, we have anthrax lethal factor. We also have the light chain and the heavy chain of botulinum. And additionally, we have this over here, which is pyroxane. So this is actually an organophosphate compound. So this is about 70% as potent as sarin.
And what will that do to you?
And this causes something called sludge, which is salivation, lacrimation, urination, defecation, gastrointestinal distress, and emesis. So you're basically pissing.
Fatal if swallowed.
you're pissing, shitting, vomiting, crying, screaming, and then you have a spastic seizure. And the way that something like this works, so the mechanism of action is really interesting, actually. So your neurons use a neurotransmitter called acetylcholine to actually support with the transmission of electrical signaling, or the transmission of general information. And what this does is generally what happens is you have acetylcholine, which is released in the synaptic cleft, so between the two neurons. And you also have an enzyme. So generally when we're talking about neurotransmitters and how they function, you'll have a neurotransmitter release, typically through a vesicle, into the synaptic cleft. And then you have an enzyme that will break it down over time. Or let's say, for example, like, are you familiar with like an SSRI?
Yeah, like Lexapro.
Okay, so an SSRI is a selective serotonin reuptake inhibitor. So what that means is it basically it prevents the neuron. So this is actually really cool. But what normally happens is your neurons release the neurotransmitter into the synaptic cleft. It should activate the next neuron in chain. And what an SSRI does is it blocks the neuron, the presynaptic neuron, from reabsorbing basically the serotonin and leaves it there for longer. So then that way you have a higher likelihood of that second neuron firing. Now, the reason this is relevant is when we're talking about something like acetylcholine, when we're talking about a nerve agent like an organophosphate compound, what it does is it binds to acetylcholinesterase. And when we're talking about esterase, we're talking about an enzyme, right? So the relevance here is that basically what this does is it binds to the enzyme responsible for breaking down the acetylcholine. So what that means is the neurotransmitter that's responsible for getting neurons to fire is left in your neurons for so long that you you go into a spastic seizure, that your muscles twitch, right? And then it causes all these secondary effects that we see that results in death, right? And it isn't like a movie where you have like this big like cloud or a smoke cloud, and you could you put on your gas mask. Most of the time you've been exposed and you have no idea, right? And the nerve agent antidotes that we use.
How long does it take to know?
5 minutes or
5
less,
minutes, okay.
right? So it really depends on the agent, but in 5 minutes you can begin to start experiencing the effects. Actually, I think you mentioned you saw the article that I wrote. In the article, I had a video of them actually applying, I think, sarin or a VX agent to a rabbit. And you can start seeing the rabbit twitching all over the place and everything, which is really unfortunate. But the nerve agent antidotes like 2-PAM and atropine, which is sort of what ATP, which is what we deploy warfighters with today, are basically Cold War era solutions. They are also therapeutics. They're not prophylaxis. And what I mean by that is they're a solution that treats or attempts to treat it. Now, it doesn't fully do that. You still need to get medical attention. It slows down the effects a bit. And actually, I think 2-PAM is reactivating, so it can sort of reactivate those enzymes and start help the body. But generally, we're looking at solutions that are applied post-exposure. So if you don't know you've been exposed,
it's impossible to do something about it.
Right. So that's why we have something. I actually have it on the table too, but we have auto-injectors. So are you familiar with?
Do you want to go grab one?
Yeah. So this is something I've used. This is something that I actually used. So I just ran the marathon, and I basically I was like, can I run a marathon with no training? Right. So I was like, what doping can I do? And how can I elevate my capabilities? So I ended up running it in an exoskeleton along with using peptides. And I actually finished.
And you have a picture of this, right? You can just throw it up in the podcast.
Yeah, I have a picture of it. But also, we have the exoskeleton over there too. You can try it on. We can maybe get a photo of you trying it on after.
Let's do that. Okay.
But point is, when we're talking about the usage of nerve agent antidotes, generally, if you've been exposed to a nerve agent, you don't want to sit there and start pulling up the relevant amount and then injecting yourself, right? So
while you're twitching and throwing up and vomiting.
Exactly, right. So you'll use something like this, which doesn't contain anything right now, but generally you'll have a syringe in here.
Okay.
And it's a spring motion that automatically injects. So it's called an auto-injector. So it automatically injects a specific dose. And you can just get these. People use these a lot for insulin.
Wait, so were you just on the trail just injecting yourself with this stuff?
But the reason is, so I was using BPC 157, which is a body protection compound 157.
What does it do?
So it's a compound that actually supports angiogenesis, which is the formation of new blood vessels, and reduces inflammation. Now, for some reason, for me, I don't know if this is a global effect across other individuals, but if you use BPC 157 or when I used it, it would basically reduce any pain I had anywhere in my legs. So, the day of the marathon at 3, 4 in the morning, I sat there injecting basically every part of my calf with BPC 157. And I had basically no pain at all during the marathon except in my feet. But it's one of those things where, I think it's 27 miles, where with no training, it was definitely harder than I thought. But I think that the exoskeleton helped a lot. The problem is that the battery doesn't support the entire run. So I had to carry 5 batteries on my back too, right? But it was one of those things that I think really did contribute. It was really exciting to see. Because
how much of the run does the exoskeleton kind of make easier?
So
is it a 15% impact or 20% impact, or?
I'll put it on you afterwards and you'll see it. They have a mode called hyper mode, and technically that thing can go at 1 horsepower. So it can go fast. It can go really, really fast. The problem is, from a battery standpoint, it can't run at 1 horsepower for the entire duration of the marathon. So I was running at 15 to 20%, which is relatively close to nothing. But I do think what it does is, and you'll try it on after, but it physically lifts your foot up and puts your other foot down. So it kind of it trolls you. It moves your body for you.
It's one of those machines at the gym.
Yeah. But exactly, it's one of those. But imagine using that to walk forward, right? So
for 27 miles.
Exactly. But anyways, my point is, for these nerve agents, generally you're going to be using an auto-injector. There's also something called a MOP suit, which you probably do want to put on screen if possible. But
sure, why not?
It's this big, clunky suit that you have to get into because you don't want to contaminate other people. And also, if you touch a nerve agent, you could contaminate yourself again. So even if you treat yourself, if it's on your hands, you can put it back into your face and you can reabsorb it or re-aspirate it or rebreathe it.
Destroy you again.
Right. So it's one of those things where you have to basically get into this MOP suit. It's this big, bulky suit. It's incredibly, there's really bad thermal management. So, you're getting into the suit and you're I know people who've been in it for 2 days at a time, right? And you're like pissing through a tube or something, but it's really gnarly. It's really bad. So
And why would you be in it for 2 days at a time? Like, what would you have to have in order to not want to?
I think that was more of a training exercise that they put them through. I think that was like an Army National Guard training thing that they did. I think if I recall correctly, but generally, if you're going, and I think if you're getting general training, you have to go into a mop suit. Even if you've seen like those videos of people in the Army going through like basic training, they put them in like that facility and they expose them to a bunch of like tear gas, right? And then they kind of leave them in there for like I think like 30 minutes and see what happens.
Really?
Well, I think they have
what
a
does
mask
typically?
on.
Oh, okay.
Yeah, I think they have a mask on, but it's still
I would hope
still
so.
very painful. So, anyways, it's one of those things where obviously we need to see the creation of, and this is something we were working on as well a while back, was a next generation nerve agent antidote. So, everything right now is, and I want to rephrase the term antidote. So everything's typically post exposure. And that seems relevant because obviously you wouldn't want to inject your body with a bunch of compounds, and then maybe not get or not encounter the threat, and then all of a sudden you have
you just have a bunch of stuff in your body, and there's no reason.
So what we were doing is, I mentioned nanoparticles before. The nanoparticles that were used in the vaccines, so in the Pfizer and the Moderna vaccine, were just like basically these lipid particles, and that's actually over there. It's an image of one, but you can think of them as like these little fatty cholesterol particles, and they can be like 1 layer, 2 layers, but generally they're very biocompatible, but they're very susceptible to like enzymes and degradation. What we were developing was this 5-layer particle, and it was not composed of fats but polymers, so plastics. And what you could do is you can actually create this particle that would more effectively get into the cells, almost taking like a backdoor entry approach. And this is through a mechanism called nanoelectroporation. What that means in practice is basically your cell membrane has like a polarity to it. And what you can do is there's a specific voltage that will actually cause a breakdown of the cell membrane and the formation of a pore. So you have to imagine like you have like this like a castle wall, right? And what you can do is you can basically shock the wall and the drawbridge falls down, right? And you can kind of have direct entry as opposed to like relying on like a convoy to like bring you in, right? So like we were like, how do we get around doing like a convoy approach, which is equivalent to your endocytosis, to a drawbridge approach, which is like your electroporation. So shocking a cell to open it, right? To like force it open for us. And we built a little nanoparticle that basically had this inner ball that would rapidly hit the different walls of the net, like the outer shell, and it would produce this electric field that would cause or basically shock the cell membrane, and it would enable like the drawbridge to come down,
forcing it to
and force it. And so that traditionally, like electroporation, like I said, in terms of the nanoparticles or the material getting into your cells for lipids is only about like 5 to 10% efficient. Electroporation is close to 95%. So it'd be dramatically more efficient in transfection or delivery of any payload. So you can be talking
like mRNA vaccines,
mRNA,
so instead of
plasmid DNA,
making it in.
Small molecule drugs, peptides, proteins, enzymes, whatever it may be, nanobodies. It could really be a wide variety of things.
I'd be curious. So I know you've got like all these different compounds and chemicals and all sorts of stuff in your lab. What are you taking regularly?
So I'm probably like a weird case study. I have generally found that like one of the things I contribute or care most about is probably my sleep, which I know we had a conversation about this earlier, and that probably seems counterintuitive on face value. But like,
do you want to just tell people like how much you're sleeping right now?
So yeah, I mean, generally don't mind if I'm not very articulate. Right now I'm on like 2 hours within the last 40 hours of work, right? But I've tried to do like different regimens around my sleep. I've tried to do the polyphasic stuff where you sleep like 3 hours, throughout the day, and then take your random naps. Right now, it's like one of those things where I don't even think it's like, I think people make sleep and make like waking up like a thing about discipline, and they're like, I need to like get up at 6 a.m. and run. And I just think that adds additional thought to your day, right? Like I'm not really somebody who aligns well to schedules. I just go to bed when it makes sense to go to bed. And generally that works very well for me. Now, I know everyone's different, but like, there's, I'll bring up Edison again, but there's like a photo which I can share with you of the guy would literally just like, he'd be working for like 40 hours straight and just get on a bench that looks exactly like this and just lie on the bench and sleep. And the only way he could do this, really, people don't know this, is that Edison was deaf. He was actually deaf, and he couldn't hear effectively. So if you actually watch videos, there's a clip on YouTube of of A Day in the Life with Edison, and you see all these people coming up right beside him, and they have to go up to him in his right ear and scream in his right ear. And initially, if you don't realize he's deaf, you're like, why is everyone just screaming right beside his face? But he was really deaf, and it's really hilarious because he created the phonograph, right? So it's one of those things where it's like, it's like the similar thing with like Zuckerberg being like a nerd, and he created the most prolific social app.
He's a super introvert.
So it's one of the it's weird. It's like being an outsider kind of gives you perspective that you might otherwise not have, right? But he's so deaf that he would just get on his bench, like literally something like this with no pillars, and just fall asleep, right?
He's so deaf he thought about hearing.
Yeah. But he actually used to say that he felt very grateful or very fortunate that he couldn't hear, because he felt that there's all these distractions, like audio distractions around him, and if he wasn't interested in a conversation, he would just pretend like not to hear it.
I know like personally, like I had real issues going to sleep for years because like even slight variations in noise level would just wake me up.
Sleep is one of those really sensitive things. It's also like one of those things where we kind of just give in. Like, I think this is probably one of the things that frustrates me the most. And even with viruses too, we treat it the same, where it's like, it's almost like godforsaken. It's like you kind of, before you go to bed, you basically submit yourself, and you hope, you're going to wake up the next day feeling good, right? But you can do very little, generally, or most people do very little to try and improve the outcome. It's not like you can go to, it's like, the parallel here is like, you can go to the gym and decide what weights you want to lift, and you can decide what exercises you want to do, and that really does impact sort of the structure and the workout that you're gonna accomplish, right? But when it comes to sleep, you're really just like
you just lay down and hope for the best.
So yeah, that was one of the things that really frustrated me, and so something that's frustrated me for a long time. It was actually one of the things that inspired the creation of the first interface that we were building, which was New Sleep, which was this brain-computer interface, or you can imagine this device that would basically shock you to sleep.
Wasn't it like a headband?
Yeah, it was a sleeping mask. Yeah. And that was also very intentional too.
Max literally talked about this when I interviewed him last. He literally talked about like using that, and he said that he felt like there was some difference.
So TAVNS, which is the modality we were exploring, or transcutaneous auricular vagus nerve stimulation, basically modulates the parasympathetic nervous system. So you might be familiar with like the fight or flight, which is like sort of like the general term that's well understood. But it down regulates the fight or flight mechanism, and kind of puts you more into like the more of the calm, relaxed state. So it amplifies the parasympathetic nervous system. It reduces the sympathetic, which is like fight or flight. And there has been some really strong efficacy on sleep. So like, for example, I think that there was like a significant reduction in the Pittsburgh Sleep Quality Index, which is one of the kind of the clinical metrics that we would employ, as well as the Insomnia Severity Index for tracking kind of the differences in sleep quality. But Brian Johnson uses VNS as well. He's a big advocate for it. Actually, I think I have the same device upstairs that he has as well. But yeah, for my sleep, like, I'll admit I've never slept on a bench before. I frequently sleep on the couch. And even beyond that, like, before bed, if I know I'm going to have a longer sleep, like for me, that's like 6.5 hours. Then I'll take GABA. I'll take a very low dose of melatonin. So like 200 micrograms of melatonin. And I'll take glycine as well.
Where do you get the 200 micrograms? Because I take I used to take melatonin, and it was 5 grams, and it was just like grabbing from Costco. My understanding is it's something like almost 100 times what you actually need.
Yeah.
Or some crazy.
I think Gwern, who's like a good blogger, has a good post on this, which I highly recommend. I'm not going to quote them. I'm not sure if it's a man or a woman, but point is, yes, you are correct. Your pineal gland generally doesn't produce anywhere close to that level of melatonin, and that's also why you experience drowsiness the next day. So to actually, generally 200 micrograms of you can procure from a lot of different places. I get it from Life Extension. That's a good source. Thorne, I don't think has that low of a dosage, but Thorne is also very good for supplements as well. I know the Superpower folks are a big fan of them. Also, I think Momentous is a newer brand that does some really good supplements. But yeah, I mean, I'll be really honest. It's like as much, or rather, the least I can think about, like the basics of living, the happier I am. So like, my mentality is like, how can I like opt for like 95% robot and like reduce like as much human
cognitive load?
Not even that, like reduce as much human characteristics as possible. Like, how can I reduce as much human need as possible? So like the need for social interaction, the need for food, the need for sleep. Like,
and how do you systematically go about reducing that?
So I don't want to mislead you and say it's systematic. It's just like, I just work. Like I don't like. It's like, it's one of those things where I've tried to like optimize my environment for like the least amount of friction possible. So like we're recording in our main lab. This is like, you know, a BSL-2 facility, broad spectrum prototyping. So we have like everything ranging from like advanced spectroscopy all the way to organic synthesis and nanomaterial and nanofabrication. I wanted to have a space where I could like build whatever I wanted to build, right? And this is really relevant because we're working on a diverse set of initiatives alongside like the federal government or like for the federal government, right? And one of the things I've tried to do is like, I think many people resort to like higher dopamine sources, so getting easily distracted on your phone. I've tried like everything from using like dumb phones, like those, you know, flip phones, all the way to like the gray screen on iPhone. I think everyone has explored that at some point. But like what I really landed on is just like, really I treat my phone like it's like almost like a dictionary. Like it just really has only books on it. I've gotten rid of like most of my social media stuff. I'm a big like I use Twitter a lot, mostly on my computer. But like I have to make myself get to my computer.
Is it like upstairs?
Yeah, it's upstairs. But
okay.
like I will like wake up and I'll like force myself to get to my computer before I can use Twitter. And by then I already want to do other work, right? So kind of right, it kind of removes the need. But again, I don't want any of this to seem like it's like discipline, because it's not that. It's just it's like motion. Like, how can I create as much motion as possible? Like the Argus sensor here, you know, I had a conversation with Delian at Founders Fund, like, I want to say 3.5 weeks ago around biosecurity. And, you know, we were talking generally about sort of the demand and kind of what it's going to take. And you know, I'd love to kind of dive up into that as well, to like actually modernize and move towards this new solution, because it isn't as simple as building new technology. But point is, you know, he came and was really interested around the concept of having, you know, some form of next generation like biosurveillance or bioradar. And, you know, basically since then it's been nonstop cracking away and building this. So it's been about 3 weeks of work, and we already have a device standing. And the next step is to do a challenge study. So
was that the like inception of it? As just having that conversation? Or?
No. So I've been thinking about biosurveillance for probably since I was very young. Have you ever played the game Plague?
Oh yeah, totally. A bunch. Were you good at it?
Yeah, so that was my favorite game growing up, or one of my favorites, at least on iPhone. And I always thought it was really compelling to like have this ability to like be this virus, and you can like infect a lot of different people. But then there's also the other side of it where you have to like make the cure, right? And it's like the other game mode you can play. And you know, I was like really compelled by this idea of being able to have something that looks like this big map and being able to see the presence of viruses at different areas across the world. And you look at the current stuff we have in place. So like the two programs, and I kind of briefly alluded to them earlier, is like we have national security. So we have BioWatch, which is I didn't fully get into it, but BioWatch was established after the American Anthrax attack. So after that army officer proliferated them, and it was part of I think it's part of Bush's one of his speeches that he announced it formally. But point is, it was in 2003 that we actually moved forward with the development and deployment of the sensors, and it was one of those things where really from the get-go there just wasn't a lot of thought put into it. So basically, we have now 3,000 sensors that are spread across the country, and we call them portable sampling units. Basically, they're these things that are about probably up to you know, your navel, so up to your belly button, and effectively are like a small silver unit and have like a little chimney-looking thing, and it basically pulls in the air, and basically viruses will attach onto the filter inside of the sampling unit. Every single day, a state or municipal level authority has to drive 4 hours to each sensor, swab each sensor, drive 4 hours back, and then they take the sample they've collected and then run it through PCR. And we're not aware of a result until roughly 72 hours later.
Which could be horrible.
Which could be catastrophic, right? Now, again, because we're using PCR for the most part, we can't detect anything emerging. So we're only detecting threats that are
known,
considered known, right?
Yeah.
And in fact, we only detect 6 of the 14 known threats. Now, I recently found out why this was the case, because obviously with PCR technology, it's advanced in the last 22 years. We should be able to detect much more than 6. The federal government, specifically the Department of Homeland Security's policy, is that BioWatch does exactly what it was intended to do, which is detect the threats that we know how to counter. So we know how to counter.
It doesn't detect the things that we don't have counters for
Exactly.
on purpose.
On purpose. So
that sounds like a horrible idea.
Now, we've tried to modernize. So the DHS's CWMD office, their Countering Weapons of Mass Destruction office, has tried to do it twice now. They had a program called Gen 3, and they had another program called Biological Detection 21. Both times, they've completely failed at determining what requirements would improve upon the current systems we have in place. But also notably, they couldn't find any technology to procure. So the kind of the standing joke with BioWatch is not even that the technology is bad or it's inundated or it's antiquated. It's that, we had a report from the Office of the Inspector General come out. So an internal report from the Department of Homeland Security where they went up to across the country and they surveyed these sensors, and they found that in 34 out of the 35 jurisdictions we've deployed them in, they don't meaningfully collect data. And they don't collect data because people unplug them. So
what?
People walk up to the BioWatch devices and literally unplug them because there's nothing in the contracts that stipulated. There's nothing that maintains that the outlets need to be locked. So people unplug the sensors and they plug in their laptops and their phones. So imagine going through TSA and just unplugging a backscatter scanner, like the X-ray machine, and just walking through onto your plane. You'd be arrested in a moment, right? If you wouldn't even be able to make it to the power outlet.
Is this one of those situations where people were optimizing for the last, the whole thing where you buy insurance for the last disaster, and because there's, there's the whole terrorist situation on the planes, they put in all the scanners and everything, invest a bunch of billions in that, and we just decided not to do it? So
generally, the federal government, when it comes to biological agents, has taken a very reactive posture. So this is what we're trying to move away from. And so the work that we're trying to do at Pilgrim, even from a policy angle, is changing the narrative that we can't wait for the next pandemic to happen for us to establish superior infrastructure. Biosurveillance and biosecurity needs to be recognized as a critical piece of national infrastructure that preserves the health and the wealth of our nation by not just detecting these threats, but if you have a system that can actually detect and attribute. So for example, we might be able to determine based off of sequencing that this specific strain is a well-understood strain coming out of a Wuhan lab, right? And if that's the case, then we're able to effectively prosecute. So that deters our adversaries from deploying potential agents if they know that if we're able to meaningfully detect and attribute the source of it, right? But we can't even do that today. So our adversaries are looking in on our current technology, and the state of it hasn't changed in 22 years. It's being unplugged, rampantly. And, we have that one national security angle. The other side of it is CDC, right? And the Center for Disease Control. And they have a program called the Traveler Genomic Surveillance Program, which is something that Ginkgo Biosecurity actually contributes to. And Ginkgo as a company is generally not doing incredibly well. But, their biosecurity division is still going somewhat strong. And they have this program called Horizon and Canopy, and part of their program involves this relationship with CDC where they do wastewater surveillance. So people also don't know this, but generally in 9 major airports, I think it's in John F. Kennedy, it's in LAX, I think it's in SFO, it's in a couple more, 9 total now.
Are they testing?
So what they do is they actually pull the wastewater off of planes. So if you use the bathroom on a plane, so if you excrete viral material, the idea is that we can pull the wastewater and then sequence or clean up the fecal matter, clean up the urine, and sequence the material and determine the presence of a threat. So actually, we just did this and we recognized that COVID is actually having another boom right now, like a summer boom, which sounds like a sale, but it's unfortunately not. It's one of those things where it sounds good in practice, but I don't know about you. I hardly use the bathroom on planes. I try to avoid using the bathroom generally when I go to the airport. So I'm not being collected. My data, my sample is not being collected. Right?
Are you doing that purposefully?
No, of course not. But it's one of those things where you might not use the bathroom on a plane. I would hope you're breathing on a plane, right? And that's where a sensor like what we're building with Argus goes much, much farther, where we can have or basically install these in airplanes or airliners and be able to effectively pull in air in real time and detect the presence of a threat.
Wouldn't an airplane actually be kind of the perfect situation, because you've got this closed environment loop where you can actually have that being measured for a very specific group of individuals?
Yeah.
Very regular. How many people fly every day? Is it 10 million?
So I don't have a firm number on that. But that's actually a good number to look into. But I would say that airplanes are definitely really compelling. I think airports as a whole is kind of a compelling first piece, and we're kind of already looking at landside deployment. So how we can work with airport administrators and kind of get into So if you think about how an airport works, you have your departures and arrivals area with baggage claim, and then you have TSA, and then everything after TSA is called clean. It's a clean area, right? Because you've kind of gone through screening. Typically, if you want to deploy something prior to TSA, or what we call landside, you don't need as much oversight. So we can deploy our sensor like Argus without having to go through the Department of Homeland Security and having to go through that whole process, which might be incredibly protracted. But anyways, we're trying to like meaningfully find deployment opportunities also in farms, like trying to detect the presence of H5N1. There's also like a really big threat of agritourism. So like recently we had two Chinese individuals who entered the country with an extremely noxious fungi that would have caused blight, like billions and billions of dollars of agricultural damage for our country, along with birth defects for people who consumed the infected crops. And they openly admitted to carrying in that fungi many times before. One of them was actively enrolled in a Chinese university. And now I think Kash Patel and DOJ is kind of overseeing the investigation. But like, you look at certain places, like you look at certain states, and many of these states are losing a wide stretch of their livestock, of their crops to a variety of different pathogens or bacteria. So there's really this prevailing issue that we need to have superior detection in place for. I think airplanes is a good opportunity. I think farms is another really clear one.
How much do you think in like damage annually is caused through preventable? Like if we just had the detection systems in place, like how much would those detection systems cost in the first place to build? And then also like what would potentially be the like ROI for on a like country wide scale?
COVID is one of those things where I think it's a really clear example of this. It's the most recent. It's sort of in like, past memory at this point. But the numbers are very clear. COVID from an economic standpoint was a $16 trillion GDP inflection, which is twice the cost of the global war on terror, which came in at $8 trillion. And obviously we're looking at a difference of 20 years versus 2 to 3 years. It also, caused, and like I can tell you pretty firmly that the source and origin of COVID was the Wuhan laboratory. There probably was some other involvement as well from other nations, but that's a whole different point. But point is, I don't know how far we can get into that.
Go right ahead if you want to.
But point is, generally, when we're talking about something like COVID, we're looking at pure casualties. If it was, let's say, intended to be an attack on our country, then from a casualty basis, it was equivalent to 412 9/11 attacks, right? So, you don't see people memorializing COVID. We obviously, I lost my grandmother to COVID. I think many people lost loved ones, but if it doesn't stand in comparison or in anywhere near foot to how we perceive 9/11, yet from a population standpoint, obviously it killed many more people.
I think probably part of the big reason for that is because you can easily pinpoint the plane crashing into the building, as you can't totally pinpoint, it's an ambiguous thing.
So what I'll say to that is, in fact, it's even worse than you expect, is that in the beginning, because we had a very limited understanding of COVID, right now the estimated casualties is like 1.23, close to 1.3 million people.
Is that just in the US or?
In the US alone. The estimations are actually dramatically higher because we've only like now isolated to like these cases that we defined as COVID related, like casualties or deaths that were COVID related. But in late 2019, kind of leading into 2020, like around December and January, there was probably a lot of people who were dying from COVID that we just didn't attribute it meaningfully to. So yeah, to your point, like, and I have different thoughts on this. I think like obviously like firm military action or like a very firm event like the fall of the Berlin Wall or 9/11 has a very clear and profound impact that sort of prevails and has an enduring impact on the country. I think we haven't truly recovered from 9/11 yet, but I think that COVID was more long stretched. I think it affected many more people. And I think from a consideration standpoint, from a defense angle, like, you look and again, people don't know this, but like for 10 weeks in Indo-PACOM, so in the Pacific, our USS Theodore Roosevelt, so one of our carriers, went down because 1,000 sailors contracted COVID. So we lost 10% of our naval air fighting capability because of COVID. And there was no direct confrontation. There wasn't a missile. There wasn't, a cruiser. There wasn't anything that hit that carrier that disabled it or sidelined it. We sidelined it purely as a result of a biological agent, right? So it kind of opens the same territory as cyber, where you can kind of have these non-confrontational attacks that have the capability set of wiping wiping the activity or the productivity of a wide stretch of infrastructure to defense, to critical resources. So it's like one of those things where clearly we need to have a system in place. And we've already spent $1 billion on BioWatch today. The CDC spent a considerable amount of money on their travel genomics program as well.
What's crazy is like even $1 billion, it sounds like a lot of money, but it's really not in the grand scheme of things. Is the goal with that piece of technology is to like kind of build the real-time plague map where you kind of see where is the plague, and you don't know, it's detecting all of the possible viruses kind of stuff?
So one thing I'll mention is, because I know it sounds ridiculous. I know the BioWatch thing sounds ridiculous, but I'm going to give them a bit of credit. And the reason I'm going to give them credit is if we detect the presence of Ebola at the Atlanta airport tomorrow, our first responders don't know how to handle that. They don't have the relevant equipment. They don't have the relevant training. They wouldn't know what to do. It would frankly be a nightmare. And it would be similar to what we were trying to do during COVID, where we'd have this massive rise of people dedicated to the problem, like we saw with Operation Warp Speed, and we tried to curb the threat as soon as possible. It did happen in 2014. There was a minor Ebola outbreak in the country. But point is, there's also a good story I'll bring up afterwards on these SAM-ridden monkeys. But point is, at that point, actually, I'll use that 2014 as an example as a clear one here. Around that time, CBP, so Customs and Border Patrol and Protection, Customs and Border Protection, was responsible for doing biological screening. So they would be asking people, oh, did you travel to the Congo? So-and-so. They're trying to determine the presence of a filovirus or Marburg virus or Ebola, like an Ebola virus. And they would also do the questions, and they would do fever screening, whatever. And basically, the CBP came forward and says, like, well, look, our men aren't trained on how to detect biological agents. We don't understand it. So let's get CDC in here. And CDC came in, and we're like, well, hey, this is interesting. And you would imagine that the CDC, the Center for Disease Control, would have a firm grasp on like disease control, but they kind of came forward and were like, we mostly do anomalies. Like we do things that are sort of out of the blue. Like for general surveillance, that's not really what we do. That's finally when DHS was kind of pulled into the mix. So the CWMD office, at DHS just got dissolved, by the way, as a kind of as a part of the Doge initiative. Now, I actually think that was probably the right move. The challenge. So one of the reasons I think largely Doge is dissolving a lot of like non-congressionally mandated offices and agencies or programs, but not agencies. But point is, one of the things we've been pushing for from a policy angle is consolidation. There is way too many. There's like 19 different federal agencies that have some level of involvement in biosecurity, and generally budgets and appropriations are highly fragmented across those, right? So like DTRA, the Defense Threat Reduction Agency, which is basically an agency inside or under the Office of the Secretary of Defense, so OUSD, responsible for curbing WMD threats, so weapons of mass destruction. And they also work with the Joint Program Executive Office for Chemical, Biological, and Radiological and Nuclear Defense. But point is, it's a long acronym. Point is, you have, I think, close to $2 billion worth of appropriations for the Chemical and Biological Defense Program, which has like 9 different offices and agencies involved. And then you also have the intelligence community. So the reason I think that the breakdown of the CWMD office was smart was largely because, in DHS alone, you had the CBRN office and the CWMD office, which really should have been doing the same thing. CBRN is chemical, biological, radiological, and nuclear. But point is, they dissolved the office and they took BioWatch and they're putting it into CISA. So CISA is the Cybersecurity Infrastructure Security Agency, or Cyber Infrastructure, something like that. And CISA, again, does cybersecurity, and now they're overseeing biological security. So the relation there is pretty challenging to identify. Now, what I've come to understand is that CISA, prior to being called CISA, was doing mostly critical security for major infrastructure. So that's sort of how they're seeing it, as like BioWatch is sort of down. Like the jurisdiction is at that state and municipal level. So that's sort of what CISA has excelled in. And
do you think it makes way more sense to have it at a federal level, like just blanket across everywhere? Or
I think it makes sense to. I mean, first off, I think one of the things we're trying to push from a policy side is accountability. I think that there's very clearly a lot of finger pointing in terms of who should be responsible for biosecurity for the nation. And frankly, it's kind of gone between public health and national security. And it really has to I think fall into probably one distinct agency in both of those categories, and then have like a unifying, joint force or unifying, not joint force, task force that would be responsible for like overseeing it, or directly involving Trump's national security advisor or the presidential national security advisor. There's a few different angles you can play here, but I think accountability is one big piece, and consolidation. I think that there's a tremendous amount of funding that can and should exist, and is incredibly distributed across these agencies and offices, and even some commands. So I think pulling it all together is sort of the right way, and I think Doge kind of breaking down some of these offices is a step in the right direction. But yeah, that's pretty much it at a high level. So just for context, getting back to my final point here is like, for us to deploy essentially like Argus, generally biosecurity, it can't be done through one sensor. So when we're thinking about a meaningful approach, even if we're talking about surveillance, we don't just have one, we don't just have like open source intelligence. We have Humint, we have SIGINT, like we have different modes of intelligence that we leverage to kind of gather a wider picture of a threat, or leverage it for an investigation, right? So my point is, like, Argus is meant to be a small piece of a much, much larger system, right? So the goal long term would be really cool to have like a plague map, but it would likely be the usage of not just Argus, but probably wastewater detection, nasal swabs. But also, one thing that I've been really interested in is, maybe there's a future where Pilgrim is responsible for privatizing like a on-the-ground force that's actually at these major airports or major infrastructure points and could actually handle threats as we encounter them, right? Or maybe we train CBP, or we train the relevant people who are already in the airport environments as part of their general training on how to handle these threats, and we provide them with the relevant equipment and infrastructure to do so. So I think that's like a second piece too, is like we can't just expect that our first responders will be sufficiently trained to handle them. We need to ensure that we can give them the relevant tools and training, and prepare them for any threat. It's like, it's almost like, the current approach is saying that, like, if it's like, we detected Osama bin Laden, but we don't know how to handle him, so let's just leave him. It just doesn't make any sense.
If you wanted to be like a super terrorist, what are the actions that you would take that you see like there's chinks in the America's armor,
Okay.
and like you can see that because you're so deep on all this stuff? Like, what are the things that you would do?
So
or focus on?
Generally, like when we're talking about like bioweapons, like the reputation is like a poor man's nuke, right? And I don't know, I think a lot of people are like now like super afraid that with like the LLMs, to like, okay, like, with AGI, we're gonna have, it's gonna be far easier to build bioweapons. If you generally know your way around a lab and you've like worked with vectors, like you've worked with AAV, like a viral vector, or you've worked with lipid nanoparticles or even polymeric nanoparticles, and you procure agents. It's like, as a laboratory, because we're not certified, I can't go out and procure Ebola, right? Ebola is highly controlled. It's mostly controlled in the U.S. by USAMRIID and CDC. But point is, like, let's say I was a terrorist and I had a source of Ebola. It's really not that tricky to encapsulate the purified virus, or just to spread the purified virus, right? Now, Ebola is a weird one. So Ebola, you can form droplets, but it's largely bodily fluids, along with fomites. So basically getting it onto a surface, and then somebody touches the surface. So like a handle, right? But like, for example, you could have a terrorist walk into John F. Kennedy Airport carrying a little tube of Ebola, spread it across the handles in all the major bathrooms, and you would never know that it was him. Like it'd be really, because there's no cameras in the bathroom, it'd be really hard set to say that this guy was the Ebola spreader, right? So
wouldn't it be like a relative, like people could trace it back after a bunch of people get Ebola in the next few days?
You'd recognize that there was an emergence of the virus, but you wouldn't know it was necessarily a terrorist attack, right? So like, that's my point, is like we have no means to actually attribute it, because we're still leveraging PCR. You'd have a lot of people who show up with Ebola, and Ebola is nasty. It turns your blood vessels into jello, which is really harsh. There's actually Richard Preston, who I just tweeted about this, but Richard Preston, who's he's a couple books, including The Hot Zone and The Demon in the Freezer, which is actually about anthrax. Funny enough, we actually have anthrax in our freezer too, so we have the demon in the freezer.
Do you want to grab that?
Yeah, I could grab the anthrax.
Grab the anthrax?
There you go. So this is what we call the lethal factor. So anthrax, and so when we're talking about things like anthrax, or we're talking about botulinum, there's different proteins, or recombinant proteins that are involved your
You may need to sit down.
Your camera guy stepped away.
I know. I saw that.
There's different recombinant.
This is the only one that Paul's been actually worried about.
There's different recombinant proteins that are involved in getting it both into the cell and then actually causing, or the mechanism that actually triggers the side effects or triggers the casualty or the eventual death. So point is, this here is the lethal factor. So this is the part of the anthrax that kills you. Now you also have the edema factor, which helps with the, I believe, supports the actual transfection into the cell. So I'm definitely more familiar on the neurotoxin side. So if we're talking about like botulinum, or people generally don't know this, but Botox is botulinum. It's a neurotoxin. And what it does is it prevents the cell's ability to actually release the neurotransmitter. But generally how it works is it uses something called the heavy chain. And the heavy chain of botulinum can basically force Paul's stepping away again.
I know.
It force the cell to form an endosome. So this is actually the anthrax lethal factor.
How much I mean, I'm not going to hold it. But how much could kill someone?
So it's a good question. This by itself would not kill you. That's why he doesn't have to be concerned, because and I'll put it away so he can come back in the room. But generally, this by itself won't kill you. You need to have a delivery system. But to your original point and what I was mentioning, I can procure this with no certification, with no license, with nothing. I didn't get verified for this. For me to purchase this, basically they called me and said, what is your experience working with anthrax? And like, what are you going to use it for? And I said, well, we work with the government, and, they checked my LinkedIn, and now's the extent of it, right? So it's one of those things where,
this is like getting approved for level 3 options on Robinhood.
Exactly. No, it's harder.
Most of the experience.
It's harder to get it's harder to get, that account on Robinhood than it is for me to procure the anthrax. And you have like 3-4 day shipping, right? And after you receive it, you can encapsulate this protein. And I don't want to give anyone ideas, but you can encapsulate a protein, this is well understood, in a vector. So a mechanism to deliver it. And if you took this and you encapsulated it in a polymeric nanoparticle, you've effectively created a bioweapon. So it's one of those things where you don't need an LLM to tell you how to do it. A polymerization, you can look at a paper on PubMed and you can figure out very quickly how to make nanoparticles. And you can procure this, assuming you've demonstrated that you have on face value a BSL-2 lab, even though a biosafety level 2 lab is not a certified lab. Like there's nobody that comes into a lab like this and says you are capable of doing it. The only thing that qualifies for biosafety level 2 is that biosafety cabinet and some general training that I'm aware of, right? So point is though that like I'll put this to the side for now. But point is like generally when we're talking about this idea of like a terrorist being able to produce a biological weapon and proliferate it, it's sort of comical how simple it is. It's sort of ridiculous. At one point in my life, I kept saying it's ridiculous we haven't seen it happen, and then we had COVID, right? So wherever you want to attribute the source or the intention behind COVID, we do largely understand it to be a lab leak. We understand that it originated in Wuhan and obviously scaled and proliferated across the world, causing global shutdowns and, millions of Americans to lose their lives. So, we're in a situation where we have seen that threat, yet we turned a blind eye. We haven't pushed for any more modernization. And the program I mentioned from the CDC. So the CDC, the country's Center for Disease Control, which should ideally be the agency or the body responsible for preventing attacks like COVID from occurring or from future pandemics from occurring. Since COVID, they were in 7 airports during COVID doing this genomic surveillance. We've now expanded to all but 9 airports. So we're in 9 airports across the country. There are 5,412 national, public airports in our country. So 9 out of 5,400 is really, really pocket change in terms of being able to stop an attack.
And are these just like the major ones like LAX and JFK?
So these are the major ones. I think the problem still relies on the fact that you might not use the bathroom on an airport or on an airplane. And frankly, if you came in from an international flight and you flew in 14, 15 hours, I don't really know anyone who's going to sit there and get a 30-minute nasal swab and PCR. You're going to want to go home, right? So it's sort of like these impractical things. I think like this is actually a really important piece that maybe we can talk a little bit more about. And it's something that I've had to contend with and spend a lot of time with is the fact that, where we are now with Pilgrim is like we're kind of the ones defining the narrative. There really isn't an organization today that serves in like a biotech prime capacity. There isn't really anyone who is trying to create this new generation of biosecurity. You have a lot of like aging inventors. I go to these events and I was telling somebody on our team, and I want to be, I'm trying to be kind because obviously there's people who've really devoted their life to this, but you have people. So you have former Undersecretary David Richardson, who was responsible for CWMD, who just got moved over to director of FEMA, going on the fact that, we can't just be thinking about the battlefield of today. We need to be thinking about the battlefield in 20 years from now. Now, if you look around the room when he's saying this, most of the people in that room won't be alive in 20 years, right? And I'm not trying to be rude, but my point is, like, we need to have this more devoted and frankly, a fringe effort to move towards changing the landscape of how we treat biosecurity today. And that really starts by creating the narrative. The piece around the narrative, which matters a lot, is positioning. So this is something I've spoken about more privately than publicly, but I think that generally the DoD does not buy the best technology. They buy the best positioned solution. And what that means is we have to spend a lot of time trying to understand not just the necessarily language, not necessarily just the language or the metrics that are relevant to the brass or to the warfighter, but also understanding procurement pathways, understanding how a product might be deployed. So a good example of this is Kingsfoil, which is our hemostatic. Initially, when I was building Kingsfoil and I tested it on myself, it was a patch, right? And I spent more time with that concept, and I realized, well, the patch design was compelling, but an IED blast, which is like a blast wound or blast injury that creates a wound, is one of the leading causes of
Would that be creation. Like a grenade?
Like a grenade or an improvised explosive device. So like a side of the highway bomb, right? And something like that doesn't create a little regular wound. It creates this highly irregular, jagged wound that looks like a shark just took a full-on bite out of your leg. So for something like that, you need something that can to treat a very diverse wound, both
like be applied in multiple places.
But also something that can fit the irregular injury, something that can fit different shapes and sizes and conditions. So like wounds also release fluid or exudate, right? So we want to have something that can accommodate a more, or at least enable a wider form factor, or broader form factor. So we moved away from the concept of like this patch towards something like a gel, right? Because that on face value seems very applicable. The problem is it's twofold. If I was to continue to try to push a gel into the Army or into SOCOM or wherever the relevant customer may be, they're trained on using gauze. So if I was to push this concept of a gel, it would necessitate training across the entire infantry. So every single infantryman, every single person in the Army who's actually going to go into a field environment, or even in reserves, would have to be trained for it.
And also, that would be a crazy thing for you to do, because obviously, like telling the Army to retrain the entire Army in order to create your shareholder value is ridiculous.
So that would never happen. So what we've done is we've established models that accommodate gauzes, right? It accommodates what we understand. So if you're trying to come in with a gel, so in fact, there was an organization, I think it's Wound Stop or Blood Stop, it's one of the two, that recently came up with this gel. Almost looks like hummus. It's like hummus in a tube, and they inject the hummus into your leg, and it instantly forms a clot. It's incredible. It's an incredible solution. They brought it to the Institute for Surgical Research in the Army, and in like the second phase of their trial, they cut it because the ISR's model relied on the fact that you have to be able to hold pressure to the wound, and because it was a gel, because it was like this fluid, they couldn't meaningfully hold pressure. So even though it's a better solution to Quikclot, and it's something, and obviously we're trying to build even something better than that, you have to be able to have something that can maintain, you can hold pressure to, something that fits within the overall semblance of what Quikclot was modeled after, right? That way you have like an apple-to-apple comparison. So if you can't do that, then they won't field your product. So my point is, like, it's these nuances and these very more esoteric bits that I've had to like procure, and like our team generally has had to build up to understand more definitively what it'll actually take to get a product into the hands of the warfighter. It isn't as simple as like an indefinite delivery vehicle, or like this outstanding contract where there's like a $5 billion budget and they can just like tap us and say, give us gauzes. It just doesn't work like that.
Okay, so I know that like there's this entire shtick in Silicon Valley where they Gavin Belson has the Blood Boy, and then it came out that like Peter Thiel has a Blood Boy, and turns out Brian Johnson now like basically has a Blood Boy, and it's his son.
Yeah.
How long until we can kind of like everyone has a Blood Boy, and what's the benefit of having one?
So blood transfusions, there's a few different reasons why people do them. Sometimes it's for platelet like changes. what I can tell you is, at least in the military context, there's a few pieces here that are really interesting. Now, obviously, there's consistently a blood shortage. That's well established. There's always a constant need for blood, and frankly, there's a constant need for human blood. Right? We in the past we've tried to give humans like other animal bloods,
like pig blood or something.
Yeah, like pig blood. And pigs are pretty close. Like swine models are pretty close for like testing hemostatic. So like when we're testing Kingsfoil, our next course of action is to basically you cut the femoral artery of the pig, so behind their leg, and you put the solution or you put the gauze on, and that's sort of how we can validate it. You can't test this on humans because, unfortunately,
if someone bleed out, it's probably not good for business.
Well, but you also can't cut holes in people's legs. So I'm very pro self-experimentation. So I did the demo on myself, largely because I didn't want to have to go through the IDE pathway or look at doing an IRB. But even what I mentioned with for Argus, right? Like for something like Argus, I intend to test that myself too. So I don't want to go through. So traditionally the CDC does something called a challenge study. And a challenge study is intentionally infecting people with a virus to then test out vaccines and therapeutics. What my plan is to do a challenge study to basically give myself H1N1 or the common cold and then cough and see if our sensor can meaningfully detect it. And then I might do a little bit of gain of function research, which means changing the actual virus and forcing a mutation, and then giving that to myself again, and then seeing if the sensor can detect it. That, or we also have the mannequin outside with the backpack. But point is, to answer your question more head on, blood is in constant shortage. It's so bad that there's a policy that people have been exploring. I actually think it has been deployed in some context in the military, where if there's somebody who's been killed, so KIA, you can go up to them and take their blood, and you can extract their blood and use their blood for transfusion with other people who are alive who are in need of that blood. Generally, the military does not carry blood because blood typically has to be cooled and kept at a very stable temperature. What they carry is something called crystalloids, which is like a saline solution, which is meant to kind of add volume to kind of help with oxygenation, but it's not a blood replacement by any means. There have been a lot of efforts to create artificial or synthetic blood, really for the past 30 to 40 years now. We have synthetic blood deployed in veterinarian contexts. So veterinarians actually do have...
For animals?
For animals, yeah.
Interesting.
So
which animals in particular?
I think mostly dogs.
Okay.
There's a company I think called Calicite, or the product is called Calicite. But the product is not very smart. Basically, it's a polymer. So it's a polymeric nanoparticle, like the one I described. And they use expired heme or hemoglobin as a means to actually carry the oxygen. So you can have hemoglobin that might be expired from a transfusion that was never delivered, and then repurpose that blood for use in what they call semi-synthetic. So that's pretty interesting. What we've been more exploring is the topic of blood conversion or blood translation, which is how we can take A negative blood and turn it into universal O negative. And it's really compelling. It's the idea of basically using enzymes to strip the antigens or the sugars off of the outer layer of the red blood cell, and basically reducing the inflammatory or the immune response that would otherwise occur from giving somebody giving you the wrong blood type.
So would it be actually giving someone that has A negative or O negative A negative, or is it just decreasing the response?
So
to getting
what you would be doing is you're literally removing the outer layer of the red blood cell that would trigger an immune response. So the reason we have antigens is for a very specific reason. Your body wants a way to detect and know if it's seeing a cell that's not inherent to your own body or seeing something that's foreign. So that's why there's antigens. Antigens are almost like a signal that your immune system can use to detect whether or not the cell that it's looking at is something that your body produced or something that's come out from a foreign source, right? So that's why people have, the different blood types is, some creatures that are 16, I think dogs have 16 blood types. It's really crazy.
Could we do gene editing to make it so everyone has the same blood type, so it's just easy transfusions?
Yeah, I don't see why not. But we wouldn't, so people have a misconception. You wouldn't want O negative blood. You want AB, AB blood. That way you have both antigens. So O negative or O blood has no antigens. A blood has A antigens, and I think B blood has.
What does that do for you?
The antigens?
Yeah.
Well, it's what I mentioned. They're the signals. Okay. So they tell the body whether or not it's a cell that's natural or inherent to your body, right? So it's a means for the body to understand and detect the presence of foreign or unknown cells.
So do people with O negative blood have like a, I don't know, slightly more susceptibility to like foreign?
It's actually what I've actually heard is the opposite. I've actually heard that people recently, I heard this actually, that people, I have not validated this, so this is
Okay.
very much in terms of scientific credibility off the record, but I heard that people with O negative blood had generally like a better response to COVID than people with A or B.
So wouldn't you want like the entire population to have O negative?
So O negative is universal donor.
Yeah.
AB is universal acceptor. So AB is what you'd want the entire population to have. AB can take anything. Because if you have A antigens, or you have B antigens, or O has no antigens, AB can take anything.
O can donate to itself though, right?
O can donate.
So in the limit, if you had everyone with O negative, it wouldn't
the
matter.
problem with O negative is that if your cells have any antigens, that triggers an immune response.
Oh,
That's why it's not a universal acceptor.
And so back to the whole like pig blood and all that, or
Yeah.
Yeah, how close are we to creating synthetic blood for humans?
We're close. It's really now come down to the FDA and sort of their purview to move these programs forward. Now, one of them has gotten like significant traction. One of them, I think, has got a big tranche of funding from DARPA to work on this. Synthetic blood is obviously of big interest for defensive applications. One of the issues with synthetic blood is actually a religious issue as well. I think it's the what is it? It's the, I'm trying to remember which religious affiliation it is.
Quran or something?
No. It's a Christian one. But I'll come back to me later on. Point is, like, part of this religion is it's going to frustrate me that I don't remember this. But part of the religion is that you can't accept like blood that is not of human origin. It's like very weirdly specific. It's not the Jesuits. It's something like that though. It's in that same ballpark. I'm Jewish, so Christianity is sort of a little bit foreign. Jehovah's Witness. Thank you.
Jehovah's Witness.
Yeah.
All right. Okay, so you mentioned like off camera that you wanted to be on a liquid diet. Is there any advantage to a liquid diet? Like what's the goal there?
It's like what I mentioned earlier. It's like reducing friction, right? Like if I've loved the idea of trying to live off Soylent. I think Soylent is like probably one of the best company names I've seen because of Soylent Green, which is incredible. That and Coneheads. But I could finally pull out the Conehead. But I'll wear it for this question. So remind me of the question. Oh, liquid diet.
The reasons for driving towards the liquid diet. Yeah.
Yeah. So when it comes to like a liquid diet, I think the general ambition is, first off, it reduces the body's energy expense on digesting food. I don't want that to like sound like I'm like overly like tracking, the energy I spend to digest everything. But I feel it's one of those things where, sometimes you eat something, it's like a bit of a rock, and that's really unfortunate. I also think there's a lot of decision making that has to go into like ordering food. So
I know the cognitive load. It's funny because if you add up all the like little decisions that you make throughout the day, like you mentioned that you don't want to have like a rigid schedule, but that almost sounds like you're making every decision like for the first time again and again and again.
I think that it's,
or are you just like having one framework, which is always do work, whatever the next step is, and
I think it's definitely more in line with that. Like I think like people like schedule because it feels like consistency to them. I've just not been somebody who opts for consistency. Like I've tried like so many times to like establish like a schedule, like, oh, I'm gonna wake up at like 6 a.m. and I'm gonna do this and do that, and then it's like, well, tonight I feel like I want to work till 4 a.m. It's like then I'm like gonna sleep 2 hours and then wake up at 6, and then my entire rest of my day is screwed. So it's like I noticed that my work schedule excels on like me being able to just switch between things. So like as it's probably very clear, we have Argus sitting there. We have Kingsfoil sitting on the bench behind it. What I've tried to do is like be in this world where I've gotten rid of like the bullshit distractions of TV. I'm a big cinephile, so I used to really love watching movies. But what I try to do now is have Argus be like a key priority. So I'm actively building like the sensor. And then if I get bored or if I feel like I'm like kind of getting out of it, I'll either move to working on Kingsfoil. So I kind of get like the dopamine hit of like working on something completely different, right? So I go from doing electrical engineering, so doing like PCB design or firmware, like firmware creation or like firmware design or just general software, to being in the lab, handling organic chemicals, and then doing or working with polymers and doing polymerization, all the way to doing mechanical engineering. And I'll kind of circle back to doing reading, right? So I kind of get to like go between these different states of working on like really compelling different ideas. And it's sort of like, I think the only way I can kind of keep sane without like having something at the end of the day to like fall on. I'm not like a big substance user. I hardly drink alcohol. It's one of those things that just like, I don't really get.
Do you only really take like the uppers, like modafinil, Adderall?
So I definitely, as I mentioned to you off camera too, like I've definitely used amphetamines. I've definitely used modafinil.
Is it a regular thing or is it more occasional?
I went through a stint where it was very regular, and I actually kind of exhausted my dopaminergic like system. Like I exhausted my ability to actually take amphetamines. So I actually can't drink any caffeine anymore. It's one of those things where if I take anything that would be an upper, it effectively puts me into a super lethargic state where I get really out of it. So I've kind of now just succumbed to being like kind of completely cold turkey and just going very natural. So it's mostly like water. I still drink Coke, so I still have like a little bit of caffeine intake, not cocaine, but which we could talk about shortly.
Save that for the next podcast.
But point is, generally, like I think my goal is, like I told you before, how can I reduce my dependency on human needs? And it isn't like, I want to emphasize this. This is not like, I'm not recommending this. This is not like a motivational podcast. And this is very much like what enables me to have the most momentum being an incredibly small team where many times I might be the only person in the lab building stuff, right? Obviously, we're kind of growing out the operation, but it's a situation where if I'm not working, then nothing's happening, right? So like one thing I've even tried to do is, I love having 3D printers, not just because of like the creative potential, but because I can have something working for me while I can sleep, right? Or I might like run a reaction and have somebody like overseeing it while I can take a nap, right? So it's one of those things where I can kind of alleviate myself while things are ongoing. And then that gives me a little bit more assurance that we're making active progress, even when I'm sleeping.
Oh, you like to see the machines kind of going in the background?
Oh, constantly.
Yeah.
Like it's pretty surprising right now. I have nothing 3D printing. Like I tried, like.
Does it cause you anxiety?
It's not necessarily anxiety, but like I don't like the idea of going to sleep without something kind of going.
You like to? Did you? Okay, so on the tangent of like, I think you used to play a bunch of video games, right? Like how much time during your childhood do you spend playing video games?
Oh God, I think that's probably why I'm a little bit weird. I was like locked in my house all day. I did not go outside. It was like, you had a friend tell me recently that she growing up, the only thing she ever did was just play Towns or whatever that game is, Toon all day, constantly, until her brain hurt. And it was very similar for me too. Like I was obsessive with Call of Duty. I played in MLG, which actually got disbanded recently, but Major League Gaming for a bit, and Black Ops 2 and Black Ops 3. And I was really big into, obviously very into Minecraft growing up.
I'm kind of surprised that it's those games instead of like the things where you design systems that are like working in the background autonomously.
So I definitely, I have to stay off Factorio because I have a dedicated group chat of friends who I get like pinged to play Factorio.
How much time did you spend on Factorio?
When I discovered it, far too much time. I mean, when I discovered Civ, I lost days of my life. Even so, like after like there was a time where, actually this leads into an interesting story where we were on this really crazy timeline to build our nanoparticle platform, the nerve agent, the nerve agent antidote, not the nerve agent, but the antidote. And this was actually the circumstance where we got kicked out of the conference, which I can touch on in a moment. But we were on this crazy timeline. I told everyone, like, look, like, I know there's Thanksgiving, the company's called Pilgrim. So you can imagine that we'd have a proper Thanksgiving dinner. And I was like, we'll delay Thanksgiving.
Just a little bit of Anthrax.
We'll do a proper Thanksgiving when we get back. Let's just keep going. And the whole team was kind of full force, working day and night, like really like almost 24/7. If I was sleeping, somebody was working. And we managed to, I have a photo I can show you afterwards, but it was basically me. I actually had to call in my brother, who has no lab experience in the slightest. He flew all the way in from Poland, which is where he was visiting at the time, like on vacation. And he arrived with his luggage and was like, okay, like, look, like, I'm like I'm like going to go back to the hotel and I'm going to settle in. And I looked at him. I'm like, no, like we're going right now. So I taught him how to use a pipette. I taught him how to use the pH meter. And in like 2 or 3 days, he was handling our cell cultures. And like he was like doing like he was basically doing the seeding of cells. He was doing the synthesis of nanoparticles. He was all over the board, and he had no chemistry or bio experience. But anyway, so
You guys were just out there doing things.
yeah, it was great. But point is, we were on this really rapid sprint, and I finally got to like a working solution. We put it into like a little tube, and it wasn't flammable, but we brought it with us on the plane, and we brought it to the event, and it looked like Kool-Aid because it had a rhodamine B, which is like a dye, a fluorescent dye, and we showed up at this conference, and I'm carrying this little blue tube, one of these tubes over here. It looks something like this, right? So the tube looks something like this with pink liquid in the bottom. I probably have one of them over here somewhere. But point is, I show up and there's like 3 4-star generals at this event, and I'm holding like a little tube of pink liquid. And we show up at the event and we go in to like present like the solution to a relevant PM. And effectively, I think the issue was that 2 people on our team weren't registered for the event, and also like they got wind of like the tube, and we were standing outside like waiting to talk to the people, like going into the boardroom, and they're like security caught us and they like saw us and they're like, we have to remove you. So while they were removing us, somebody on our team ran into the conference room, and there's a person there like from the agency. It was the JPO, so the Joint Program Executive Office, and told the guy like, look, they're kicking us out. Can we present to you outside? And this was like a brand new conference center in Florida. The only thing outside of it was just the garbage bin. There was not even tables. It was still under construction. So we basically get this guy to come outside, and like in like a Shark Tank style pitch, we have him put his computer on the garbage bin, and we're all crowding around him, pitching him this nerve agent antidote. And I'm holding up like a pink vial, right, of like solution. Anyways, this is all to say that it was in a crazy, crazy sprint. We got kicked out by armed security. And after that, this really insane sprint, I spent like a day and a half straight on Civ. I just basically hunkered in my room, and it was nonstop. But it's one of those things where you get to see the creation of an empire. You have like different systems level thinking, and then you get to like a result much quicker than the type of work that I do in the lab. So it's sort of a little bit of a cheat code. But
what do you think in the limit the Jake Adler Pilgrim empire is going to look like?
It's a really good question. I think that I've tried to be cautious of overstating or estimating sort of the eventual impact. I think like there is a tremendous room for what we're doing not to work, and like for this thesis to be wrong. And that's like actively what I'm trying to prove every single day. Like I know like where we are. Like we're still very much early. Like the team size and our facility is definitely in line with like a day 0 company. Like we're still in the beginnings here. I think we have a lot of clarity and context. Like I think one thing I've been really opting for is to like move towards understanding reality. I think like beyond building a really generational company or something that's an enduring institution, I think I'll be satisfied if I like walk away feeling like I actually understand how things work. Like what can actually like enable me to like kind of play with the fabric a bit. But I think I've said publicly, the goal in one vein is to establish a biotech prime. I think that there is a really big vacuum for that. But it really, like, if you think about what we're trying to do, it's the same. The thing I said at the beginning of the podcast is like, how do we more meaningfully take concepts that may have tremendous impact on the patient and get them to them sooner? And I think the military context somewhat trips people up because they're like, are you building bioweapons, or why are you doing trauma care? It's like sort of a little bit odd when people think about the context of drone warfare and like this Deus Ex Machina of like removing the human from the equation, which of course will never happen. But my point here is that I think that the DoD really serves as like a really strong first market for us to take meaningful solutions and field them in environments that are highly austere and fitting to the capability sets that we're interested in pursuing. But there's no reason why hemostatics or biosurveillance or electronic tourniquets or blood conversion or synthetic blood or bone mending tools or a variety of the other things I've thought about shouldn't exist in a civilian oriented context. I think like there is a tremendous potential here, not just to build Biotech Prime, but ideally the most valuable medical device company in the world. And I think like there is precedent for this. I'm non-traditional. I didn't graduate school, but like you look at somebody or an organization like Medtronic, which obviously is the leading provider of pacemakers today, and it was a guy named Earl Bakken who started that company, and the guy had no experience with medical devices other than he would actually repair medical equipment. And in that process, he familiarized himself in the Twin Cities with a lot of the doctors. And then there's this instance where, I swear to God, people probably don't know this, how pacemakers were actually created. He was looking at a magazine. It was an electronic magazine for an electronic metronome. And he built the circuit. And I think in the following days, there was this big blackout event in the Twin Cities that resulted in the NICU, like the neonatal intensive care unit, going down. So basically losing all their power. And a lot of the kids, or a lot of the fetuses, or whatever you want to call them, can't really call them fetuses at that point, but a lot of the babies were on like some variant of like life support. And what he did is he took this electronic metronome and they just implanted it. They, like, with no trials, no clearance, nothing. He literally went up to the baby and just plopped it right in. And this wasn't like a pacemaker like you think about today, like a little thing. This was like wires, like electrodes coming out of the baby's chest, going into like a refrigeration size unit, like a household refrigerator, right? So it was one of those things where, the guy it was very non-traditional. He came in from this unconventional lens. So same thing with Frank Hersey doing hemostatics. I think it kind of takes an outsider to look in on the system and understand sort of the bureaucratic inertia and the entrenched interests. And I think generally me being in a position where I've been synthesizing technologies and taking a very generalized approach, taking a very Edisonian approach to creating solutions and systems, is very fitting of the type of technology and solutions we need, not just in the military, but the civilian market as well.
Did you study anything on like the history of like the Nazi Germany's labs and how they kind of operated? Because I imagine that they, for better or worse, had lots and lots of people that they were able to test things on, whereas that's not legal today.
Yeah, so this is something that I think is sort of, I'm not going to say is prevalent to the same extent today, but China definitely has lower ethical constraints, and there's already been, I think, a former Secretary of State or former CIA director came forward. I think it might have been John Ratcliffe, came forward saying that China has openly tested and done CRISPR on soldiers. So they've already moved forward in that world. But to answer your point directly, I mean, human enhancement has always been an area of interest. The Nazis also used to do like a lot of, they established like a whole science around measurement. I don't know if a lot of people know this, but they would have very specific tools that they could use to like measure the distance between people's ears, the color of hair. They would almost have like a swatch panel of like all the different colors of hair, and they would compare it to people's hair color to see whether or not they were a Jew. So they had all these different like techniques and approaches that they used to like create like this Jew science to like track down and isolate like the people who might be Jewish and in hiding, right? But my point is, like, obviously Nazi researchers and scientists had an abundance of test subjects to work on. Now, Nazi Germany was less involved in the biological side of things, from like a biological warfare component. They were much more involved in chemical weapons. Like they did a lot of production of chemical agents. But they tested on twins. They tested on a variety of different subjects. But yeah, I think like generally, like, from a human performance enhancement standpoint, they also used Pervitin. So Hitler was an ampet user of Pervitin, which is basically an amphetamine tablet, which could also be made into a solution. So that's sort of why Hitler, there's been like the videos of him tweaking a little bit. And
you said that they were like, were they also putting that in chocolate? Yeah.
So there's something called like Coca-Cola, which is like it's like Coca-Cola, but I think show. And it's basically a chocolate with caffeine and cola nut. So actually, you might have seen this in like grocery stores. Have you seen like the Awake candies?
Oh, yeah.
So it's sort of like that. I think that's maybe where they got the inspiration from. But there's been reports of them spiking the chocolates with amphetamines. So, you would have a warfighter who would be in a highly austere condition or on the front lines consuming a chocolate that they might rather think is just caffeine. And all of a sudden they're spiked with something equivalent to Adderall or Dexedrine, right? So obviously...
So the soldiers didn't even know that they were taking it?
It's probably unlikely. Yeah, I mean, it's very unlikely that there was that level of transparency that existed. And it's one of these things where, I don't want to go too deep or too pedantic here, but like technological supremacy is something that I think is largely overrated. I think it's something that we've been really pushing for as a country. because it seems like it's the right narrative. But we look at like Vietnam as an example, and we bombed the Ho Chi Minh Trail, thousands of times, and we hardly disrupted it. We had vastly superior technology to the Vietnamese and the Viet Cong, yet we lost that conflict. Or we look at something like the Germans, where in World War II, there was after World War I, there was an embargo on armored vehicle production. So the Germans weren't able to produce or modernize tank designs until Hitler became chancellor. And the Brits and the French specifically the French, had incredible tanks. From an air standpoint, the Germans were superior, but from a land standpoint, the French were vastly superior along with the British. And my point is, like, you had an environment where the French would be maintaining the front line, or would have these checkpoints, and then you had German troops leveraging a blitzkrieg strategy, kind of going around them. But also, blitzkrieg was really just rampant Germans. It was basically Germans who were probably high as shit on amphetamines, and instead of stopping, what they would do is they would attack a unit, they would take over the position, and they wouldn't like stop and rest. They would just keep fucking going, right? So, I really, unless they had like this crazy level of conviction in the party, you have to assume that there was some level of usage of amphetamines or uppers as
using chemicals to kind of change brain
exactly.
State. Yeah. Interesting. Okay, let's end on what's the hardest thing you overcome?
Mm-hmm. Generally, the companies people are building today, and I'll just be very honest, exist in the narrative that somebody else created. So, Anduril with Palmer and Trey and Brian and Matt have done a phenomenal job creating this new narrative of the implications of autonomy and drones in a warfighting environment. You had Palmer Luckey going up on stage, and it wasn't an offhand compliment or offhand comment. He would say, the John Deere tractor's a better AI than our military. And, the point there is driving frustration through, revealing absurdity, right? And I think my point is, like, there is sort of this narrative that a lot of organizations today in the drone space are a derivative of. A lot of companies are kind of following in line with the kind of the piece that Anduril set down. But really, it feels like what we're building here at Pilgrim is not just, I've somewhat repudiated our association with that narrative, and I felt that we're very much in the job of myth making. Like, we're in the position where I have to kind of create the right narrative and positioning and products that can meaningfully take my core thesis and philosophy around leveraging the DoD as this on-ramp to like unload and kind of rapidly expedite the development and clearance of medical devices into a tangible organization that's driven by a shared mission and ideology. So I think my point is, like, from a standpoint of things I have to overcome, it's one of those things where nothing ever feels easy. Like, almost every challenge that I think does exist, I've had to, every challenge that exists in these earliest stages, I've had to encounter, ranging from even thinking I could do all the work myself to realizing that, my goal is to make myself somewhat more obsolete and bring on more people that can kind of alleviate some of my responsibility, to fundraising and being super conservative, to not spending enough money on equipment and then realizing I have to spend 30 hours or 150 hours repairing a scanning electron microscope. I feel like I've really had to go through this process of creating and defining a narrative around what we are building at Pilgrim, but also being the person boots on the ground who's actively involved in nearly every component of development today. I have really every part, from a policy, from a DoD, from an FDA, from a development standpoint, kind of circling through my brain. And obviously we have to scale this, but I think my point is, we're still very much in the beginning. I think we're still deeply misunderstood. I tell our team a lot that I think it's a little bit feels like O5 Palantir. It's one of those situations where there's probably a long stretch ahead of us for us to establish the right organizational structure and the defining product that kind of carries us into this next phase. But where we are today is I am all in. I've been consistently all in, and I've noticed that with a level of divine intervention, or rather divine spirit, I've been able to follow, or rather the term here is divine providence, is I've been able to kind of follow my own will, and it's led me closer to the results I want. So I think generally overcoming narrative, overcoming positioning, and creating incredible technologies.